basically related with conduction of the sound waves. Then there is a middle ear cavity, which is connected to the nasopharynx through this pharyngeal tube. And then we have the inner ear that is actually situated inside the petrous part of the temporal bone. All these three parts, they develop from three different structures, and the most important part, then they actually develop separately, then they make a connection with each other, and finally they make one entity. uh, Break in union of any of these three parts will lead to congenital deafness. Okay, ये एक आप जरा जहन में रख लें कि congenital deafness क्यों हो सकता है, क्योंकि किसी भी part में अगर defect हुआ तो it will end up in the congenital deafness. One part is inner ear. If that is not developed, there will be deafness. If the middle ear cavity is not developing properly, again there will be no communication between the external and the inner ear, and there will be conductive deafness. अगर external ear ही नहीं है तो definitely the sound waves will not pass, and it will end up in the congenital deafness. ठीक है? The other important point you must remember is that the first part among all these, the external, middle, and the inner ear, the first part which develops is the inner ear. Then the middle and the external ear, they develop all together, while inner ear is the first structure which actually develops. So this is the middle ear cavity. It's a box-like structure which has the four walls, and then it has the roof, and it has the floor as well. It consists of different structures. Especially, you must know that there is the ossicles, which are the smallest bones situated in the body, and then there are muscles. The two important muscles are stapedius and the tensor tympani. There are ligaments, there are the nerves which are crossing the middle ear cavity, especially the seventh cranial nerve, facial nerve, and one of the branch. They are lying inside their own bony canals. They are not lying within the cavity, they are crossing it. And another is the chorda tympani. These nerves cross, and whenever there is middle ear cavity infection, it is possible that they may involve the facial nerve, or they may involve chorda tympani as well if the infection is very deep. Plus, the communication of this middle ear cavity is posteriorly towards the mastoid antrum. So whenever there is middle ear cavity infection, they can involve the side of the skull in the form of mastoid involvement. They can be involved secondary to the pharyngitis or tonsillitis, and then they are connected via the malleus to the tympanic membrane, and via the oval and round window to the inner ear. So the infection of the middle ear cavity as well as the external ear, they are actually dangerous because they have direct communication with the brain. Then we have, I hope you all remember how they are directly communicated with the brain. The petrous part of the temporal bone opens into the posterior cranial fossa via internal auditory meatus. Then, okay, this is the inner ear. I hope you all know very well there is an organ of hearing that is called the cochlea, and then posteriorly there are three semicircular canals which are related with the valence. Both of these are connected via nerves, that is the vestibulocochlear nerve, which has two parts. The vestibular part is related with the equilibrium, and the cochlear part is related with the hearing. These two parts, along with the facial nerve, they leave the facial nerve enters, and vestibulocochlear leave the internal auditory meatus. Okay, now this is the cut section. Why am I revising this? The reason is that during development you must understand, if I talk about bony labyrinth, and you must understand what is the membranous labyrinth. Okay, so these membranous and the bony labyrinths, they are situated inside the petrous part of temporal bone. The membranous labyrinth is actually membranous structure which is lined by the epithelium. The bony labyrinth also is lined by the epithelium, but it develops from the mesenchyme, and different types of fluids are present. Can anybody tell me what is the difference between endolymph and perilymph? Yes, anyone? What is difference between the endo and the perilymph? Ji, Noor Fatma. Can you explain what is difference between endo and perilymph? There are two Noor Fatmas, I think. 63 is present. Roll number 63. Ji. Okay, Muhammad Salim.
Miss?
Ji. You are
Miss
Noor Fatma?
No, Miss Tuba.
Okay, Tuba, yes, good.
Miss, endolymph is present in the membranous labyrinth and it has a higher composition of potassium. And
Very
perilymph is present in the bony labyrinth and it has a similar composition to that of ECF.
okay, good. Endolymph is present inside the membranous labyrinth and perilymph is present in the bony labyrinth, right? And they have different mineral composition, ionic composition rather. The endolymph is high in potassium and low in sodium, and the perilymph is high in sodium and low in potassium. This difference helps in the ionic movement along the hair cells, that is called as the organ of Carti, or the also they are helpful in maintenance of the balance, where they stimulate the crista ampullaris. These are the sensory organs for the balance as well as. Very good, Tuba. Thank you so much for your answer. As far as the so I just wanted to if you know the proper gross anatomical structures, then you are able to understand how the development is taking place. So this hole is actually a bony canal. This bony canal contains inside the cochlear duct, which is called the membranous labyrinth, and in addition to that, it also contains the bony labyrinth. The bony labyrinth is divided in two parts. One is the vestibular canal, the other is the tympanic canal, or they are also known as scala vestibuli and scala tympani. Okay, and this in the center or in the middle is the cochlear duct. Now, this cochlear duct is separated from scala vestibuli by vestibular membrane. Okay, and this scala tympani is separated from the cochlear duct by the basilar membrane. Okay, and the organ of Carti lies on the organ of Carti or Carti, whatever you say. It consists of the hair cells, and these hair cells have a membrane which is known as the tectorial membrane. Now, the movements of perilymph induce the movement of the endolymph, and that endolymph causes movement of the tectorial membrane. So when the tectorial membrane is lying over these hair cells, tectorial membrane actually pulls the hair cells. Okay, when they pulls the hair cells, there is ionic exchange. There is movement of the ions, and ultimately a person is able to hear. And this movement in the perilymph and endolymph is induced by the waves which are transmitted through the external auditory meatus, the sound waves, which cause vibration within the tympanic membrane, and this vibration causes movement of the handle of malleus, and this handle of malleus will move the incus and the stapes. Stapes is connected to the oval window, which then moves uh, what we call as the endolymph, the perilymph and the endolymph. Okay, now this is the whole process how we are able to see. Okay, now this is how this is tympanic membrane. I hope you can see the arrow. Tympanic membrane moves, and this causes movement of the malleus, the incus, and the stapes. And this is the foot place of the stapes, which cause movement of the fluid in the perilymph as well as the endolymph. When there is movement of the perilymph, it causes movement of the tectorial membrane, and ultimately the hair cell. You can see this is the reddish is the tectorial membrane, to which the hair cells are attached. These processes, when they move, they cause the ionic exchange. Okay, this is vestibular apparatus. That was about organ of Carti or Carti, and the vestibular apparatus is there are three semicircular canals. Okay, and at one of the, and there are 5 openings within the utricle. Okay, I hope you all are clear about saccule and utricle. Saccule is connected to the cochlear duct, and the utricle is connected with these 5 semicircular canals. Now, at each end of the semicircular canal, there is a dilatation. You can see here the dilatation, which is called the ampulla. Ampulla contain the structure that is called the balance organ or sensory cells for the balance, and these are called crista ampullaris. Now, these are stimulated. In other words, the vestibular canals, they are as if the structure, like, you know, you have seen the jalebi that contain the outer crust, and inside there is a fluid. So it's like a fluid moving inside. So, and this responds to the movement of the head or positional changes in the body. This balance is actually in response to the positional changes in the body. You move your head, but you don't feel vertigo. This is because your balance is maintained inside the semicircular canals. Now, development of the inner ear. This is all about the gross anatomical structures, and then it's not, you know, much detail is not required as far as development is concerned. But at any point, if you are unable to understand, you can share your question in the chats. Meanwhile, or at the end, I will ask you to, you know, I'll give you the time for the query. Remember that the development, I told you that there are 3 different units of the ear: external, middle, and inner. But the first portion which develops is the inner ear. Now, how it develops, and remember that at first, the development of ear is in the neck region. They are quite low because neck is not developed. So the location of the ear is as if in the neck, and then there is extension of the ear with the growth of the face and with the growth of the pharyngeal arches. So the first sign of appearance of the ear is called the ear placode. Now, what is the ear placode? Ear placode is thickening of the ectoderm. फुरकान, ये जो एक मुझे नजर आ रही है स्क्रीन, प्लीज मूव दिस विंडो अवे. आपको नजर आ रही है?
यस मैम.
ये क्यों आ रही
मैम
है?
आप स्लाइड जो है दोबारा से शेयर कर लीजिए, तो मेरे ख्याल से फिर चली जाएगी. स्टॉप शेयर करके दोबारा से शेयर कर लीजिए.
अच्छा अच्छा, ठीक है. क्लोज करके दोबारा शेयर करूं?
नहीं, ऊपर देखें स्क्रीन के टॉप पे जो ना स्टॉप शेयर लिखा होगा, रेड कलर के बटन होगा एक सेंटर में, वो क्लिक करके दोबारा शेयर कीजिए इसको.
ठीक है, थैंक यू. ओके. अच्छा फुरकान, ये रिक्वेस्ट आ रही हैं पार्टिसिपेंट्स की, तो मुझे क्या एडमिट करना है उन्हें?
मैडम नहीं, वो मैं देख लूंगा.
आप देख लेंगे? ठीक है, ठीक. ओके ओके ओके. नाउ on day 22nd we are discussing development of the inner ear. On day 22nd, I hope you are aware about this structure. This is the heart tube, developing heart tube, and this is the future head region of the embryo. This is the first pharyngeal arch, and this is the second pharyngeal arch, this purple. In between these pharyngeal arches, there is a cleft. Okay, and when we look from inside the embryo, if we cut this embryo, at the side of the cleft there will be pouch. Okay, so pharyngeal pouch, pharyngeal cleft, and then above and below would be the first and second pharyngeal arch. Now, this is the proper area where ear will finally be located. But the appearance of inner ear is actually a thickening within the surface ectoderm, and this forms a thickening which is known as otic placode or ear placode. This otic placode is a thickening of the surface ectoderm which forms a vesicle, and that vesicle then detaches from the surface ectoderm and is implanted inside. So basically the inner ear is derived from the surface ectoderm, okay, not from the neural tube or other structures. Do not get confused with it, because it's written that there is with the development of the neural tube and closure of the neural tube, it induces the surface ectoderm to form the otic placode. It induces, it does not form the otic placode, remember it. So it's important that neural tube must form and close, and it will induce the formation of the ear placode on the sides of the neck. If there is no formation of neural tube, there will be no formation of ear. So this is the pit which is called the otic placode. Now how this pit changes into inner ear? Okay, this bluish color you can see, this is the first and the second pharyngeal arch being developed, and this is the bluish pit which is the future ear. So it is quite low near the cervical somites. Later on it will. Now here is what I wanted to tell you, the importance of the tube. Now this is the tube which is about to close, and this is the surface thickening of the surface ectoderm, which is called the otic placode. This otic placode forms a pit. This pit then finally detaches from the surface ectoderm and goes inside. And around that, the petrous part of the temporal bone will form later on. So first there is formation of soft structures, then there is formation of the hard or the bony structures. Okay, this is another diagram. This is the development neural tube. This is notochord, and this is the thickening of the surface ectoderm and thickening of the surface ectoderm, plus this is the otic, and finally it detaches from the surface ectoderm and forms the otic vesicle. Okay, it is a continuous. Aman, please switch off your mic. Okay. There is thickening of the surface ectoderm, then there is formation of the pit, and then this pit gets detached from the surface ectoderm. This is called the otic vesicle. Okay, this is the first step in the formation of the inner ear. How this otic vesicle is converting into the ear, this is what we are going to discuss. And remember that this has nothing to do with the middle ear or with the inner ear. Oh, sorry, the external ear. So inner ear is entirely a separate entity. Now when this otic vesicle develops, this is the circular structure, and this is surrounded by this pinkish color structure which is all mesenchyme. So it is developing within the mesenchyme. Now when this otic vesicle is formed, then slowly and gradually it elongates. Okay, it elongates and forms one ventral portion and one dorsal portion. This is the ventral portion and this is the dorsal portion which is going behind. Another important structure I want to tell you is that the otic vesicle or the otic pits, they are formed at the level of rhombencephalon. Okay, they are formed. Why they are formed at the level of rhombencephalon? Anybody? No? They are formed at the level of rhombencephalon because the nerve, which is, what is rhombencephalon? It is hindbrain. And where is the situation of a vestibulocochlear nerve? It has to come from the cerebellopontine angle. It is situated within the pons. Okay, so this ear has to be connected to the rhombencephalon or the pons. That's why it is situated over here. And then it is with the expansion of the head and formation of face, the ears are finally located at the sides of the head at the level of starting from eye up to the depends on the length of the ear. So this otic vesicle, now what is the sequence? There is thickening of the surface ectoderm, then there's formation of otic pit, and then there is formation of otic vesicle. Now, when there is formation of the otic vesicle, it then divides into a ventral portion and a dorsal portion. ठीक है? Ventral portion क्या बनाएगा? The ventral portion will form the saccule. The dorsal portion will form the utricle. ठीक है? Saccule से फिर cochlea develop होगा and utricle से क्या develop होगा? Semicircular canals. Again, come to this picture. This is the ventral and the dorsal division. The ventral division is saccular portion and the dorsal division is the utricle portion. First, we are discussing the ventral portion, that is the development of the cochlea. ठीक है? This is the otic vesicle or ear vesicle, which is detached from the surface ectoderm, and this has divided into a dorsal portion and a ventral portion. Now, concentrate on the ventral portion. Projection will go on increasing in size within the mesenchyme. Just keep in mind that all around this vesicle, there is mesenchyme. So this duct has to develop within the mesenchyme, and it is important that this mesenchyme should remain there. The reason is that the duct is developing from the otic vesicle, and otic vesicle is developing from the surface ectoderm. This mesenchyme is developing from the neural crest cells, and this mesenchyme will give rise to the bony labyrinth. Okay, so membranous labyrinth is developing from surface ectoderm. Bony labyrinth will develop from the mesenchyme, which is surrounding the membranous labyrinth. First, there will be development of membranous labyrinth, then there will be development of the bony labyrinth because of induction of the mesenchyme by the membranous labyrinth. The language may be a little difficult for you, but it's important to understand. So this is the cochlear duct, which slowly and gradually grows and makes 2.5 turn. Okay, this growth is as if you have watched the cartoon Cinderella. The fairy changes the carriage, the pumpkin into the carriage. Okay, there is the growth of the or formation of the wheels of the carriage in this form. So slowly and gradually, like a sprout, the cochlear duct is coming out and making 2.5 turn. This 2.5 turn will form the cochlea. Now, during this formation, there is all around the mesenchyme. This mesenchyme will form the bony labyrinth. Okay, so this is the development of the saccule and the cochlea. Okay, this is the cochlear duct which has developed. Up till now, the organ of Carti has not developed. Organ of Carti has not developed. This is the mesenchymes. These white cells are the mesenchymes surrounding the cochlear duct. Now, slowly and gradually, these the cochlear duct cells, they divide into two parts. One is known as the outer ridge, another is known as the inner ridge. Okay, outer and inner ridge. The outer and inner ridge will form the hair cells. The outer ridge will consist of the 2 or 3 rows of the hair cells. Okay, 2 or 3 rows of hair cells. Inner ridge will consist of only 1 row of the hair cells. Now, these hair cells will be covered by tectorial membrane, and this is known as the organ of Carti, and this is connected with the cochlear part of the vestibulocochlear nerve. I hope it's clear. The formation of the cochlear duct and then the formation of inner and the outer ridge, which further differentiates into 2 or 3 layers of the hair cells, which are very specialized cells, and there is formation of the tectorial membrane, and there is connection with the cochlear part of the vestibulocochlear nerve. Now, during formation of this organ of Carti, the outer mesenchyme then divides into membranous labyrinth, but there are bony labyrinth. There will be 2 parts of the bony labyrinth, one which is above and one which is below. Okay, so this is the organ of Carti, and this is the one part which is above and one part which is below. The part which is above is known as the scala vestibuli, and the part which is below is known as the scala tympani. Scala vestibuli is separated through vestibular membrane. Scala tympani is separated through the basilar membrane, and these will contain the perilymph and the cochlear duct will contain the endolymph. I hope the development of cochlea along with the membranous and bony labyrinth is clear to you. Okay, this is the inner and the outer ridge, and how you can see the cells are connected to the cochlear part of the, each cell will contribute to one nerve fiber. Okay, each cell will contribute to the one nerve fiber, and all these nerve fibers, they will combine and form the nerve. This is the organ of Carti. This is how the hair cells are actually attached to the tectorial membrane. Okay, when tectorial membrane moves, it pulls the hair cells. When it pulls the hair cells, there's exchange of the ions. Then we are coming to the development of the utricle. As I said, utricle is the posterior part of the otic vesicle. Okay, the anterior or ventral part of the otic vesicle was saccule. The posterior or dorsal part is the utricle. Now, in the utricle, there are three. You can see this is the single, but later on it divides into three separate canals. These three separate canals will ultimately join together, grow, and form the three semicircular canals. These three semicircular canals, again, they contain this forms the membranous labyrinth. They would also be surrounded by the bony labyrinth, and they will contain the one part of the canal will dilate and form the ampulla, and that ampulla inside will contain the crista ampullaris that will respond to these are the sensory cells which will be connected to the vestibular part of the vestibulocochlear nerve. I hope it's clear to you. That is the formation of the inner ear. Okay, and this formation of the inner ear is the first part to develop, and this develops during the 22nd day. The formation of the cochlea and utricle or semicircular canal happens during 6th week. Okay, so this inner ear is formed on day 22nd. A vesicle is formed on day 22nd, and the utricle, cochlea, semicircular canal, they start forming during the 6th week. The organ of Carti is formed during 10th week. Okay, 9th and 10th week there is formation of organ of Carti. Now, after this inner ear, there is development of the middle ear. Now, middle ear, remember that inner ear is developed from the surface ectoderm. Middle ear is developed mainly from the first pharyngeal pouch. That is the, this is the, you might be remembering the development of the pharyngeal arches, pouches, and clefts. As far as cleft is concerned, only first pharyngeal cleft is important. Rest of the clefts, they are superimposed or covered by the rapid growth of the second pharyngeal arch. ठीक है, दूसरे clefts normally पहले सारे clefts होते हैं, first, second, third, fourth, but later on all these clefts will be covered by the second pharyngeal arch. Okay, they have to disappear. Otherwise, if they do not disappear, they will form the sinus. So this is the, you have to concentrate for the ear development at the first pharyngeal pouch and cleft. So this is the, where I have kept the arrow, this is the first pharyngeal cleft that will contribute to the external ear, and that is part of the external auditory meatus. The first pharyngeal pouch will give rise to eustachian tube as well as the middle ear cavity. Now what is happening here, you can see the external appearance. This is the, here is the future otic placode. This is the first pharyngeal arch, which is also called the mandibular arch. And this is the second pharyngeal arch, which is supplied by the facial nerve. The first pharyngeal arch is supplied by the mandibular branch of the trigeminal nerve. This is the third, fourth, and then there are fifth and sixth pharyngeal arches. Now what is happening, how there is development of the middle ear. I told you middle ear cavity, eustachian tube, they are developing from the first pharyngeal pouch, but the ossicles and the muscles, they are developing from the arches. The first pharyngeal arch, okay, I am coming to the ossicles later on. First discuss the development of the cavity. There is expansion of the pharyngeal endoderm posteriorly towards the ear. Okay, this is the pharynx whole region. From here, the first pharyngeal pouch extends towards the ear to form the middle ear cavity, to form a cavity-like structure that is connected, that will make two connections. One connection is with the nasopharyngeal tube, another connection would be to the external auditory meatus. But till this point, there is no communication. They will just be opposed. This is the first pharyngeal pouch, and this is the first pharyngeal cleft. First pharyngeal cleft will move inwards to form the external auditory meatus. First pharyngeal pouch will move posteriorly, okay, until these both communicate with each other. That the point of union is the tympanic membrane. So tympanic membrane would basically be a combination of first pharyngeal pouch and first pharyngeal cleft. This point is very important to understand. So coming back, this is the first pharyngeal pouch, and this black you can see is the first pharyngeal cleft. They both are growing towards each other. Ultimately, they will make a connection. Okay, so this the first pharyngeal pouch gives rise to the tubotympanic recess and the tympanic cavity, but the ossicles lying inside and the muscles lying inside the middle ear cavity will be developed from the first and second pharyngeal arches, not the first pharyngeal pouch. So this is again another diagram where you can see this yellowish is the pouch, and this outside, this is the cleft. Ultimately, they both will join each other. The point of union is the tympanic, the future tympanic membrane. Okay, now there is development of the ossicles. So from where the ossicles are developing, the ossicles are actually developing the malleus and the incus. They are developing from the Meckel's cartilage, that is the cartilage of first pharyngeal arch, and the stapes is developing from the Richter's cartilage, that is the cartilage of second pharyngeal arch. Okay, these questions are asked: what are the derivatives of the pharyngeal arches, pouches, and what is Richter's cartilage, and what is the Meckel's cartilage, and what is the nerve of first pharyngeal arch, second pharyngeal arch. That's why the reason that why facial nerve supplies the stapedius muscle and why the trigeminal nerve supplies the tensor tympani is the reason, because tensor tympani is related with these two, incus and malleus, and the stapedius is attached to the stapes. That's why facial nerve supplies the stapedius muscle, and the tensor tympani is supplied by the first pharyngeal arch nerve, that is the mandibular branch of the trigeminal nerve. Clear about this? I repeat how there is development of the middle ear cavity. The cavity is developed from the first pharyngeal pouch. It grows posteriorly, and this connection which grows posteriorly, actually it is coming from the pharynx. So that's why this narrow channel will be known as the eustachian tube, or the tubotympanic, the eustachian tube or the auditory tube, and this tubotympanic recess is the posterior expansion of this first pharyngeal pouch that will form the middle ear cavity. The ossicles will be developed from first and the second pharyngeal arch. Okay, so after that, there is development of the external ear. This external ear development is, there are two structures which are forming the external ear. One is the external auditory meatus, the other one is the pinna, okay, pinna or ear or auricle, the external ear. So this external auditory meatus is developed from the first pharyngeal cleft. While development of pinna is separate, we'll be discussing in just a short, after a short while. So this, you can see, this is the tubotympanic recess. This is the bluish is the first pharyngeal cleft. Slowly and gradually, they both will communicate, and this is covered or is blocked. The external auditory meatus is blocked by the meatal plug that will be then dissolved, and there will be canalization of the external auditory meatus. So this is the first pharyngeal arch which is forming the cleft. This is the first pharyngeal pouch, and in between there is mesoderm. That's why there are three layers of the eardrum or the tympanic membrane. And tympanic membrane, someone may ask you how tympanic membrane is developing. Its inner layer is developing from the first pharyngeal pouch. Its outer layer is developing from the first pharyngeal arch or ectoderm of the first pharyngeal arch. Its middle layer is developing, that is the connective tissue is developing from the intervening mesenchyme. ठीक है, तो ये दो क्लेफ्ट और पाउच आपस में मिलते हैं और बीच में सैंडविच हो जाता है mesenchyme. So in this way there are three layers of the tympanic membrane. The inner one is the first pharyngeal pouch, the outer is the epithelium of first pharyngeal pouch, outer one is the epithelium. The outer one is the epithelium of the of the first pharyngeal cleft, and in between there is mesenchyme. Okay, now this was about development of external auditory meatus. Then there is development of the pinna or the auricle. Now the auricle is a very complicated structure. ठीक है, complicated क्यों है? क्योंकि इसमें different parts हैं, concha, then tragus, then helix, all these different, how they develop at the site of in between first and the second pharyngeal arch. First and second pharyngeal arch. In between, 6 swellings appear. 3 on the front, 3 on the back. ठीक है, ये आप देख सकते हैं. These are the 1, 2, 3, 1, 2, 3. And what is the site? In between first and second pharyngeal arch. In between first and second pharyngeal arch, there are 6 swellings which appear to form the pinna or auricle. This is the first pharyngeal arch, 1, 2, 3. That's why the ear, pinna of ear has dual supply. ठीक है, facial nerve की branch भी supply करेगी and the mandibular or the trigeminal की branch भी. Now all these 6 ear hillocks, इनको कहते हैं 6 ear hillocks. They modify, they grow, and ultimately you can see they form a ear which is finally giving the shape of the ear. Each and every person has a unique ear. Now the problem is that the most congenital structural abnormalities which arise within the ear are related to the pinna. आपने देखे भी होंगे, हो सकता है आप में से स्टूडेंट्स में किसी को अगर हैं, you know, there are appendages like जो ऐसे कभी कभी ट्रेगस 2 में डिवाइड होता है, इसी तरह जो ये एंटी ट्रेगस है वो 2 में डिवाइड होता है, या एक्स्ट्रा अपेंडेज होता है. आई विल शो यू द डायग्राम्स. बहुत कॉमन है क्योंकि ये 6 कभी कभी प्रॉपरली ज्वाइन नहीं कर पाते. ट्रेगस की जगह पे अगर 2 जो हैं इन्होंने ज्वाइन नहीं किया तो दे विल रिमेन सेपरेट. ठीक है, सो दैट्स व्हाई दे अल्टीमेटली एंड अप इन द ईयर appendages. So the development of auricle is at the site of first and second pharyngeal arch in the form of 6 ear hillocks. Now coming to the congenital abnormalities, I hope you are clear about the development of the inner ear, the middle ear, and the external ear, right? So congenital deafness, as I said, that since there are 3 different parts which are developing, failure of communication between these 3 parts can end up in the congenital deafness. If there is only conducting part is deficient, then with the ear, you know, prosthesis and the ear apparatus, we can restore the deafness. Okay, if the inner ear is properly developed, if there is a bit of middle ear cavity, even if the external ear is absent, we can, you know, do the plastic surgical operations and ultimately implant the auditory implants and hearing implants, hearing aids, and the baby may be able to hear. So for the congenital deafness, there are many, you know, causes, genetic causes, or most of the time it is syndromic. एक movie भी आई थी Treacher Collins syndrome पे, Wonder, in which there is a boy with, you know, lots of abnormalities, facial abnormalities. That was Treacher Collins syndrome, in which there is small ear and then rest of the abnormalities. So congenital deafness can occur. Apart from that, there are other abnormalities. For example, anoxia. Anoxia means there is absence of the external ear. Okay, absence of the pinna or auricle and the external auditory meatus, means there is defect in the formation of first pharyngeal cleft. Maybe there is defect in the formation of inner and middle ear as well, but here there is development of, and see the micrognathia as well. There is small lower jaw as compared to the upper jaw. Okay, microtia means small ear. Something is there. You can, I told you, 6 ear hillocks has to join in a complicated fashion to form the ear. But by some reason, if these hillocks remain separated, do not form proper ear, either it will end up a very small ear or abnormalities like this. There may be a normal ear, but some pits are there, which are actually communications through the first pharyngeal cleft. Now this these are known as pre-auricular pits. Okay, इनका वैसे तो कोई नुकसान नहीं है, but sometimes they act as a sinus and continuously discharge होती है. For that, this has to be removed surgically. Then appendages, ये मैं आपको बता रही थी, these are very common. Okay, if there is failure of union of the auricular hillocks, or if there are extra auricular hillocks, they will end up in appendages like this. Okay, these does not cause any, you know, structural problem. Just a cosmetic reason if one person wants to get it removed. Just a summary of what we have discussed: the inner ear, the middle ear, and the external ear. They are all together derived from different structures. The inner ear is derived from the otic vesicle, which in turn is a structure that is derived from surface ectoderm. This divides into secular and utricle portion. The secular portion forms the cochlear duct, and the utricle portion forms the semicircular canals. Then there's the middle ear, which is formed from the first pharyngeal pouch, as well as the arches, and the external ear, which is formed from the first pharyngeal cleft and the 6 auricular hillocks. Now, any question if you want to ask? Yes.
Excuse me, ma'am.
Can you please tell me your name, beta?
Miss tympanic membrane एक बार please दोबारा explain कर दें.
ठीक है. Who is this?
Zunaira.
Spelling?
Zunaira.
Zunaira.
Yes,
Zunaira.
miss.
Okay. Yes, Zunaira.
Miss tympanic membrane एक बार explain
जी
कर
जी
दें.
जी. Since tympanic membrane, आप यूं समझ लें कि जैसे दो कमरों के बीच में अगर एक दरवाजा हो, ठीक है, सो दरवाजा इज अ पार्टीशन इन बिटवीन 2 रूम्स. सो ऑन वन हैंड जो एक रूम है वो इनर ईयर है, जो बाहर की तरफ रूम है वो है आउटर ईयर. इन बिटवीन दरवाजा इज द टिम्पेनिक मेम्ब्रेन. सो टिम्पेनिक मेम्ब्रेन बेसिकली, और मैंने आपको बताया कि जो मिडिल ईयर और इनर ईयर, ये 2 डिफरेंट स्ट्रक्चर से, इवन इनर ईयर भी डिफरेंट स्ट्रक्चर से डेवलप हो रहे हैं, लेकिन फाइनली इनको एक यूनिट की तरह काम करना है. देखें, अगर यहाँ से वेव्स जाएंगी तो डेफिनेटली टिम्पेनिक मेम्ब्रेन के थ्रू उनको मिडिल ईयर क्रॉस करना है और फिर इनर ईयर में जाएंगी. So what is happening as if my right hand, this hand is pharynx or pharyngeal endoderm, and this left hand is the external part. This is forming the external ear and this is forming the inner ear. This is the first pharyngeal pouch and this is the first pharyngeal cleft. अब क्या होता है कि slowly and gradually ये दोनों आपस में मिलते हैं, ठीक है. तो अगर बीच में कोई स्ट्रक्चर आ जाए, ये मीजनकाइम है बीच में, ठीक है, तो टिम्पेनिक मेम्ब्रेन इस तरह बन जाएंगी. ये मीजनकाइम ऑलरेडी बीच में है, यहाँ से फेरिंजियल पाउच आ रहा है और यहाँ से फेरिंजियल क्लेफ्ट. तो नाउ देयर आर 3 layers of the tympanic membrane. What are those? The outer epithelium is formed by the first pharyngeal cleft. The middle portion, that is the connective tissue, is formed by the mesenchyme, and mesenchyme which is present there at that area, and the inner layer epithelium is formed by the first pharyngeal pouch. So there are 3 layers of the tympanic membrane and they are formed from the 3 different structures. What are those structures? First pharyngeal pouch, first pharyngeal cleft, and the intervening mesenchyme. This is the structure, ठीक है. अंदर से फर्स्ट फेरिंजियल पाउच आएगा और ये बाहर से फर्स्ट फेरिंजियल क्लेफ्ट जाएगा. जहाँ पे ये दोनों मिलेंगे वहाँ पे थोड़ा सा मीजनकाइम भी होगा जो कनेक्टिव टिश्यू बनाएगा, और वहाँ पे एक मेम्ब्रेन बन जाएगी which would be attached to the handle of the malleus, and that handle of the malleus is or malleus is derived from the first pharyngeal arch. ठीक है, तो ये सारे interconnected structures हैं. Then it forms a thin membrane जो साउंड वेव्स की वजह से मूव करेगी. ठीक है, ये यूं इस तरह साउंड वेव्स की वजह से मूव करेगी, और यहाँ पे हैंडल ऑफ मेलियस अटैच है, तो हैंडल ऑफ मेलियस फिर मूव होगा. हैंडल ऑफ मेलियस इंकस से अटैच है, इंकस मूव होगी, इंकस के साथ स्टेपीज अटैच है, स्टेपीज के साथ अटैच है पेरीलिम्फ, तो पेरीलिम्फ मूव होगा, वाइब्रेशन होगी पेरीलिम्फ में, तो द कॉक्लियर the basilar membrane and the tectorial membrane will move, and ultimately it will be converted into the sound sensation. Is it clear, Zunaira?
Yes, ma'am. Thank you.
You're welcome. Any other question?
Assalamu alaikum, ma'am.
Walaikum salam, beta. Who is this?
Ma'am, Ushna.
Okay, Ushna. Yes, please.
मैम आप मेम्ब्रेनस लैबिरिंथ की दोबारा से एक्सप्लेन कर देंगी डेवलपमेंट?
Formation?
जी, फॉर्मेशन.
Okay. Now, membranous labyrinth is actually the main structure of the inner ear, right? Now, this membranous labyrinth is formed, if I start from the very beginning, that is the first part of the ear which is developing during day 22nd. By day 22nd, I mean that 7 3s are 21, right? Starting of the 4th week. Day 22nd is the starting of the 4th week. There is a thickening of the overlying surface ectoderm. किस जगह पे थिकनिंग होगी? जहाँ पे rhombencephalon अंदर डेवलप हो रहा है. ठीक है, प्लीज बी अटेंटिव टू दिस डायग्राम उष्णा. दिस इज द न्यूरल ट्यूब. ओके, ये न्यूरल ट्यूब का वो पार्ट है जो प्राइमरी ब्रेन वेजिकल बना रहा है. कौन सी वेजिकल? rhombencephalon. ठीक है, इस जगह पे because of some genes which are released from the rhombencephalon, they induce the surface. वरना आप पूछ सकते हैं कि भाई जहाँ पे mesencephalon है वहाँ पे भी ईयर बन जाए, जहाँ पे prosencephalon है वहाँ पे भी ईयर बन जाए. नहीं, the genes are released from the area where there is rhombencephalon. So these stimulate the surface ectoderm. ठीक है, ये surface ectoderm वैसे इस तरह होना चाहिए, smooth. But now इसका कुछ पार्ट, अच्छा पूरा भी यहाँ से यहाँ तक भी ईयर नहीं है, एक छोटा सा पार्ट सरफेस एक्टोडर्म का थिक हो जाएगा जिसको हम कहेंगे ऑटिक प्लेकोड. वो फिर इन्वेजिनेट होगा अंदर और एक स्वेलिंग की तरह बना देगा एंड अल्टीमेटली डिटैच होगा. ठीक है, अल्टीमेटली इट विल डिटैच फ्रॉम द सरफेस एक्टोडर्म एंड दिस इज नोन एज ऑटिक वेजिकल. फिर सरफेस एक्टोडर्म ऊपर जो है वो रिपेयर हो जाएगा. ठीक है, और ये ऑटिक वेजिकल अंदर चला गया. तो पहले क्या डेवलप हो रहा है ईयर का स्ट्रक्चर? ऑटिक वेजिकल. नाउ दिस ऑटिक वेजिकल इज गोइंग टू फॉर्म द मेम्ब्रेनस लेब्रिंथ. तो अगर आपसे कोई पूछे कि मेम्ब्रेनस लेब्रिंथ इज ड्राइव्ड फ्रॉम न्यूरल क्रेस्ट सेल्स, सरफेस एक्टोडर्म, फर्स्ट फेरिंजियल पाउच, फर्स्ट फेरिंजियल क्लेफ्ट, इट इज एक्चुअली द सरफेस एक्टोडर्म. ठीक है, ऑटिक वेजिकल, ऑटिक पिट, ऑटिक प्लेकोड, मेम्ब्रेनस लेब्रिंथ, all are derived from the surface ectoderm. So this otic vesicle will change into, आपको ये नजर आ रहा है पीछे से, ये आगे से थोड़ा dilated है, पीछे से constrict हो गया. So it is dividing in two parts. The anterior part would be known as saccule, the posterior part is known as utricle. अब saccule जो है, that saccule and cochlear duct will be formed from the ventral portion of the otic vesicle. Utricle and semicircular canals would be formed from the posterior or dorsal portion of the cochlear duct. Again, this is the diagram. Then there is slowly and gradually expansion of the membranous labyrinth. ठीक है, the anterior portion forms saccule. Saccule से फिर एक process निकलेगा which is known as the cochlear duct. This will go on increasing. पहले first turn होगा, एक टर्न पूरा होगा. ठीक है, फिर दूसरा टर्न पूरा होगा, फिर आधा टर्न पूरा हो रहा है. अब देखें ये आधा टर्न है. ठीक है, that's why we say cochlea has 2.5 turns, 2.5 टर्न, राइट. अच्छा, ये फिर कॉक्लियर डक्ट बन गई. उसके बाद फिर ये जो पीछे यूट्रिकल है, that will divide into 3 main parts. These 3 parts will elongate in the form of 3 semicircular canals. अब जब ये मेम्ब्रेनस लेब्रिंथ बन रही है उसी टाइम पे आउटसाइड मीजनकाइम होगा. This mesenchyme will change into the bony labyrinth. ठीक है, this one. sorry, I'm quite ahead. This one. This is the cochlear duct. Initially it's a single tube. उसके बाद इसके सराउंडिंग जो मीजनकाइम है वो चेंज होगा और वो दो पार्ट्स में डिवाइड होगा, एक कॉक्लियर डक्ट के ऊपर और एक नीचे. एक बनाएगा जो ऊपर है वो स्केला वेस्टिबुलाई और नीचे जो होगा कॉक्लियर डक्ट का, दैट इज एक्चुअली द बोनी लैबिरिंथ. The bony labyrinth is actually it is obliterated initially. Then there is vacuolization. Vacuolization की वजह से उसमें cavities होंगी इस तरह, ठीक है. ये cavity बन जाएगी स्केला वेस्टिबुलाई ऊपर और ये स्केला टिम्पेनाई. जो बीच में है ये कॉक्लियर डक्ट है. अब कॉक्लियर डक्ट में फर्दर मॉडिफिकेशंस किस तरह हो रही हैं? दैट इज द मेम्ब्रेनस लैबिरिंथ. इट विल फॉर्म two rows of the two different parts of the epithelium because they have to form the organ of Carti. The organ of Carti is formed from the inner and the outer ridge. ये outer ridge है और ये inner ridge है, ठीक है. ये दोनों parts फिर cover हो जाएंगे tectorial membrane से and they will form the special hair cells and they will communicate with the cochlear part of the vestibulocochlear nerve. So your question was that how the membranous labyrinth is developing. It is developing from the otical vesicle, otic vesicle. Otic vesicle is dividing in two parts, utricle and saccule. Saccule is forming, further dividing into the cochlear duct, which is then connected to the saccule by the ductus reunions. आपने gross anatomy में पढ़ा होगा, saccule is connected to the cochlea through the ductus reunion. ठीक है, ductus reunion is this part. I'm showing you the ductus reunion is this part, okay? क्योंकि ये finally extend होगा and ultimately ductus reunions will be connected to the saccule and cochlea. ठीक है, उसके बाद फिर उसी के दौरान ही ये this happens during 6th week. Otic vesicle 4th week में बना, 6th week में utricle और saccule, cochlear duct and vestibulocochlear canal बन जाएंगे, vestibular canal. The organ of Carti is formed usually during 9th to 10th week. Is it clear, Ushna?
Yes, ma'am. Thank you.
You're welcome. Any other question? No question? Okay, let me just